So in this segment, we're going to be talking about the scientific method. And I know what you're thinking. It's just a bunch of steps that we have to memorize, we have to talk about in every science class. And I guess that's partly true, but the scientific method is just an organized, common sense approach to solving problems, to answering questions. And if we're about anything in science, it's about answering questions, understanding the reasons why things happen the way they do. That's really what science is all about. So science, all scientific discoveries begin, the very first step is an observation. Simply observing something in nature, something that's happening in front of us that we don't understand, that we can't instantly explain. Why does it happen that way? What's making it happen? And so a good example of that, one of my favorite examples, one of the greatest medical discoveries in the 20th century, happened in 1929. A microbiologist by the name of Alexander Fleming accidentally introduced some mold onto his the Petri dishes that he was working with. You see this white substance down here is a type of mold. And what he was trying to do was to grow bacteria. And you can see this sort of zigzag pattern that you see on the Petri dish is bacteria. And anyway, he noticed that whenever mold was growing on the Petri dish, that there was always a zone, an area around the mold where bacteria did not grow. It was an observation. The bacteria does not grow anywhere near the mold. And that observation caused Fleming to start asking some questions. And that's what we do as scientists. We get curious. Well, why does it happen that way? So he wondered, why is the bacteria not growing near the mold? He wondered, is the mold producing a substance, a protein perhaps, that is killing the bacteria or preventing them from growing? And most importantly, he wondered, could that substance, could that protein produced by the mold be used to kill bacteria in our bodies? If we have a bacterial infection, could we use this substance made by the mold to fight that infection and to kill the bacteria? The name of the mold, by the way, is Penicillium, and the substance produced by the mold, of course, is penicillin. And it was the discovery of the first antibiotic. It all started with an observation. So in science, an observation leads us to ask questions. And the basic question that you're trying to answer is called the problem. So observation and then defining the problem is the first step of the scientific method. Now, if you've ever studied this before, and I know you have, I know you've been taught the scientific method before. You may have been taught that after the problem, that the next step is hypothesis, but there's actually a step in between. Remember, the hypothesis is supposed to be an educated guess. It's a likely correct answer to the question. And so if it's going to be an educated guess, you first have to gather information. You have to do some research. You don't just willy-nilly randomly guess what you think the answer is. So that research and gather information step is really important. After you have a hypothesis, which is a possible, maybe even probable explanation for your problem, you then must test that hypothesis with an experiment that involves recording data. And then you're going to analyze that data to form a conclusion. Now, when I say form a conclusion, that always refers back to the hypothesis, because the conclusion is where you say, where you decide, okay, the data clearly supported the hypothesis. We're going to accept our hypothesis based on the data. Or you say in your conclusion that the data did not support the hypothesis, and therefore we're going to reject that hypothesis. Apparently, it is not the correct answer. And that's okay. That does not equal failure in science, because once you realize that your hypothesis is not correct, you simply circle back to the research step. You gather more information and you form a new hypothesis. And then you test that. And you keep circling back until you finally find a hypothesis that is supported by the data. And once you have that, then the last step is to publish your results. That is to share what you've done and how you did it with other scientists. Because if you've only conducted one experiment that supports your hypothesis, that could be a fluke, So what you would like is you would like other scientists to replicate your work, to conduct the same experiment in their own laboratories. And if the experiment is repeated many, many, many times by many, many, many people, and the data supports the hypothesis every single time, it's probably the right hypothesis. And that's where we start getting into the area of what we call a scientific theory, a hypothesis that has been tested and tested and tested and has a lot of data supporting it. In science, we refer to that as a theory. So a theory is not just a hunch or something that's maybe true. In science, a theory is something that's already been tested and that has a lot of data supporting it. So I thought it might be useful at this point. I know you guys know about the scientific method, but I thought it might be useful to walk through an example. And so this is just totally hypothetical and maybe a little silly, but let's suppose you made an observation that crop plants, let's say bell pepper plants, grow better in western North Carolina, in the mountains, than they do in central North Carolina. It's just an observation. Maybe they grow faster, maybe they produce more crop per acre, more profitable, for that reason. So that's an observation, and what it causes us to do is to ask a question, right? Define a problem. So the question or the problem might be, is there some substance, some nutrient that is in the soil in western North Carolina that's not prevalent in the central North Carolina soil? Obviously, there's some climatic differences, so I guess you'd have to eliminate those as possible explanations. But let's say you in your research step, when you're gathering information, you discover that western North Carolina soils are rich in magnesium, which is an ion that is present in soil sometimes. And you do some research and you discover that central North Carolina has a much lower quantity of magnesium in the soil. So you develop a brand new fertilizer product. And you think this is going to be even better than Miracle-Gro. We're going to call it Ultra-Gro. And our hypothesis is Ultra-Gro, because it contains large amounts of magnesium, will grow healthier plants. They'll grow taller, produce more peppers. And of course, we need to test that hypothesis. Now, if I'm going to test the hypothesis, I'm going to set up two groups of plants, or at the very least, two different pepper plants, both the same species. And I'm going to have some constants. That is, I'm going to have to treat these two plants identically as much as possible to eliminate as many variables as possible. So I'm going to grow them in the same type of soil. Maybe it's not soil that would be found in western or central North Carolina. Maybe it's a store-bought potting soil. I'm going to give them the same amount of water and the same access to sunlight. We call these experimental constants. So there's really only going to be one difference in how I treat these two plants. And that one difference is called the independent variable.